Okay, recording has started going on. Good. To change the slide, right?
Yes.
Okay.
Yeah.
All right. Let's get started. Again, I'm really happy to see so many faces before Thanksgiving. Nobody started celebrating Thanksgiving too early. That's good. Thanks to all for coming. Actually, I'm very happy to say have Professor Guillermo Aguilar coming from Texas A&M. That's where I got my PhD from. So it's actually the department head of my alma mater here. So it's really my pleasure to invite. But I've known Guillermo for a long time because we both work in heat transfer, but not necessarily in the same areas. But we used to bump into each other in conferences when we were assistant professors, and then of course we had our careers cross paths many times. So as soon as the pandemic let up, and I said, well, it's time to bring him here and show him how great NC State is, right? So he's here. He's currently the department head, and also James and Ada Forsyth professor, and the department is also endowed as J. Mike Walker's 1966 Department of Mechanical Engineering. Professor Aguilar got his undergraduate in Mexico, and then he got his master's and PhD from Santa Barbara, UC Santa Barbara. And then he was a postdoctoral fellow at UC Irvine. And then he joined UC Riverside in 2003 and quickly rose through the ranks and became the department head, where he served for 8 years as department head before he made the transition to Texas A&M in 2021. He works, as you see, in the area of bio heat transfer, and that's his a major area. And he's done work in lasers and surgery, and works in various areas: NSF, NIH, AFOSR, Sandia National Labs, all kind of funding from everywhere. Research interests are in cryo spray cooling, laser tissue interactions, biomedical optics, and materials laser processing. 100 papers, 50 plus graduate students, and certainly continuing to do research as. Welcome, Guillermo.
Thank you, Srinath, and thank you all for coming. I also didn't realize that this was a week prior to Thanksgiving, so I knew it was a risk to plan it this way, but it seems that it worked out. Thank you so much. Well, as Srinath just said, I am the department head for Texas A&M, and let me wear that hat for a little bit before I actually get into the technical presentation. That's in my job description. I have to do it. So let me tell you a little bit about our department. We, like Srinath just said, we have a very large department. This year we are at 75 tenure track faculty and 23 APT, what we call APT, which are academic professional track faculty. So that's a combination of instructional research, professors of practice, this kind of faculty. So we're pretty close to the 100 mark. That's very large. Obviously, we need about 30 staff, or a little over 30 staff, to manage a department of this size, with those many students of 1,500 undergrads and 530 grad students. Roughly, the grad students, roughly half and half, are masters and PhDs, and the 1,500 undergrads are just sophomores through seniors, so he doesn't consider the Common Core freshman students. We have been growing for a while. As you can see in this graph, we've been adding faculty over the years. I just came in 21, but right off the bat, I had the fortune of hiring 10 wonderful faculty members. 5 of them joined us just this year, 3 assistants and 2 associate profs, and 1 more will join us in the fall of 23. Those are the 6 tenure track, and then the ones with the maroon color at the bottom, those are part of the APT group, different titles. So hiring 10 in 1 year, that was, you can imagine how big of an enterprise that was. We're very proud of the awards and recognitions that we give to our faculty, and this is just a sample of the latest ones we have either internally or externally we have awarded to them. In fact, I think most of these are external. And Srinath, I'm pretty sure you recognize a lot of faces there. So we're very proud of this, and we know we have high quality faculty. So. Oh yeah, it was before. Yeah, these are just the latest ones. They keep accumulating these awards year after year. I told you about the almost 1,500 undergrad students. This is the way in which they have been growing. The past few years, as you can see, we haven't had a lot of growth at the undergraduate level, which is very welcome and very fortunate for us because we really needed to catch up. We were having issues of student to faculty ratios that were getting up there. So we stabilized the enrollment of undergrad students, and now it's something more manageable. Just like you, we're very proud of the kind of accomplishments that our undergrad students achieve year after year. We just took 1 first place for a 3rd consecutive year in the SAE Aero. This is a multidisciplinary enterprise for us. We don't have the aerospace engineering as part of mechanical. They're a separate department, but students integrate to work on these type of projects, and they compete really well, as you can tell. We're already competing in the SAE Formula Electric, which is relatively new. And we continue to do a lot of capstone courses like the one that is shown here about the crown that goes on the Queen Theater in Bryan. You probably remember that. And so we're proud of these kind of achievements as well. Lots of scholarships and fellowships that we award year after year, thanks to the support of mostly our former students or alumni. We call them former students, who give a lot of scholarships and fellowships. And every year we honor them and we give them out to many students. Graduate students continue to grow. As you can see, there's more than 500 now, and a pretty even distribution between PhDs and masters, different types of masters, but master students nevertheless, which keeps these very vibrant research programs going. And so finally, we are obviously very proud of the recognition we're receiving from our peers. I'm pretty sure you do, as well as we do, keep track of the U.S. News and World Report type of recognitions. One thing that I came in and realized is that they were only focusing on U.S. News and World Report, which, as professors know, that's essentially based on a perception type of assessment. But the Shanghai rankings, which are a little more quantitative, were not actually considered. So yes, we compare ourselves all the time with other public universities and institutions like yours with the overall ranking, and we feel pretty proud of where we stack up, and obviously we're aiming at doing better. But with the Shanghai rankings, we're really scoring quite well. That's as you can see, there's 5 different types of assessments that they make based on high impact publications, citations, etc. And there we occupy the 4th place worldwide, which is pretty high, but it's also first in the US. So we're very proud of this recognition as well, and we're making sure that we convey that to our peers. Just to wrap it up, there's another grant that I'm pretty sure you know about, the NSF RED grant, which stands for Revolutionizing Engineering Education. We're one of the recipients of this grant, and we have been implementing this for a couple of years. The main purpose of this grant is to transform the curriculum and beyond. We're focusing more on the culture of our faculty who deliver the curriculum, more so than making incremental changes to their curriculum themselves. I would need another seminar to explain to you all the details about it, but just the take-home message of this is we're convinced that making changes to the curriculum is important, but it's even more important to make changes to the culture that the faculty in the department have in terms of delivering the curriculum to our students, because that's more long-lasting. All right, so I was already introduced, and I'll just walk through this very quickly. I was born and raised in Mexico City in particular. I was fortunate enough to attend the largest university of Mexico, which I think still is the largest in the world, with 350,000 students altogether. I don't think there's anyone close to that. I was there in the late 80s and early 90s, and then shortly after that, I went, like Dr. Eckert said, to UC Santa Barbara, beautiful place. If you've never been there, I recommend it. It's a beautiful location. I spent there 6 years working on my master's and PhD, and then I went to UC Irvine, where I worked at the Beck. It's hard. And my office was facing the ocean, just to give you an idea. It was hard, yes. UC Irvine is not bad itself. You know, I then moved there, and a beautiful location, not quite on the coast, but only 15 minutes away from the coast. Another beautiful location. I spent there 4 years working, with various positions, but ultimately I was housed at the Beckman Laser Institute, which is one of the premier institutions that develops laser technology for medical procedures. And then I went to UC Riverside. I was just hopping UC campuses, I guess, and I went to my third one, another beautiful campus, but that's a little bit more inland. And I spent 18 years there. I was fortunate to develop my career at UC Riverside. I was the department chair for 8 years, and then Texas A&M came and snapped me and showed me that I could continue my career there. So that's where I am. All right, so this is briefly the outline of what I'll tell you about in terms of my technical work. I'll give you a very brief overview of what I did during those first years as an assistant prof, and then I would like to focus more on the last two topics there related to a project that we have entitled Window to the Brain. It was supposed to end in 2020, but because of the COVID, we had to extend it, so it's closing next year, 23. And then I'll also tell you a little bit about some of the cryopreservation technologies that we're developing under the NSF ERC auspices. So when I first started at UC Irvine as a postdoc, I was introduced to this kind of dermatologic conditions. I didn't know anything about it. I was a mechanical engineer, and like I suppose most of you, we don't normally see these kind of things from a technical standpoint. And I was even wondering, why would they hire me to work on something like this as a mechanical engineer? And little by little, I understood that there was a lot that they didn't understand about heat transfer processes in biological tissues. And so I was fortunate enough to work with Dr. Stuart Nelson, who's one of the premier leaders in the development of technology that treats these kind of conditions with laser surgery. So I'll briefly describe what this is. Well, you can read here, these are vascular malformations that are typically located in the dermis, about 100 to 500 microns underneath the skin surface. And they don't affect a large number of individuals, but the few that they affect can develop some psychological conditions if they're not treated early on in life. So Dr. Nelson's whole career has been dedicated to trying to remove these stains particularly from children as soon as possible, so they don't have to undergo these psychological affections. Thanks to the development of laser technology, what we can do nowadays is to shine a laser of a specific wavelength that is preferentially absorbed by the hemoglobin of blood. And that generates enough heat, and that's where the mechanical engineering analysis comes in, generates enough heat so that it photocoagulates, that's a technical term we use, photocoagulates the vessel. And by doing this systematically, we can remove the excessive number of vessels that give place to that coloration. The problem is, and here's where they they were facing some issues back then, that there's another chromophore, this is another absorber that is sitting in the epidermis, actually in the junction between the epidermis and dermis, and that's the melanin, right? So unfortunately, patients that have higher concentrations of melanin, meaning darker skin types, they're less susceptible to the benefits of this kind of technology. And so the two problems that they were facing back then was that there was an excessive amount of epidermal heating that could lead to some scarring and dyspigmentation, and also, of course, that serves as a filter, so less light was being delivered to the target. They had already developed in the mid-90s a technology of cryogenic spray cooling. So Dr. Nelson and his co-workers had already patented, actually, this technology. It was very simple. It was just based on spraying mist of this very cold liquid on the skin with the purpose of developing a thin layer that it would evaporate and extract the heat from the epidermis shortly before the laser was fired, with the intention of making it further down to photocoagulate the vessels. So this was great. It was protecting the epidermis the way they wanted it to be. It was allowing for higher energy dosages, which in turn reduced the number of treatments. And it was also helping as an anesthetic kind of effect, because the cold usually removes the pain that sometimes happens especially with high laser power when you shine it onto your tissue. Like I said, they patented this back in the mid-90s, and this became one of the most profitable patents in the UC system, even to this day. So great, it was good for them. I arrived too late to that party, so I didn't get any benefit from that. But I had the opportunity to work on improving the technology of cooling. And so this is just an example of the kind of patients that Dr. Nelson would see, and the kind of outcome, therapeutic outcome that he was obtaining when he had access to fair-skinned patients like this child, right? No filtering at the epidermal level, because melanin is very, very little, so it was an excellent result. But the vast majority of patients who had even mild levels of melanin concentrations were not reaching that same level of improvement. So here's really where my work started, and for the 4 years, the last one of last century plus the 3 of this new one, I started working on cryogenic spray characterization. And that entailed a lot of different things. So for example, I went too fast. We had, had to study the internal flow to see what kind of two-phase flow was taking place within the nozzle as this liquid was flowing through those nozzles, then characterizing the sprays in terms of droplet size distribution, velocity, temperature, also looking at how that was impacting on the skin and what kind of mechanical deformation this spray was inducing on skin, because that led to an entirely different type of heat extraction. And to do all this, we would actually use some materials that had thermophysical properties similar to skin to measure the temperature profile underneath the surface. So a bunch of epoxy and agar gels and these kind of models that we used to simulate that skin tissue that we were cooling with these sprays. These are some of the pictures of those systems that we developed over time, trying to understand also the radial variation in temperature when those sprays were impinging on the skin and spreading away, et cetera. And then measuring, like I said, the temperature and heat flux that was induced by a variety of different nozzles that were aimed at a different distance away from the skin, because that also had a big effect on the temperature of the sprays, all to basically develop an optimal system that would lead to the maximum possible heat extraction from the skin. So that was basically a lot of what I was doing at UC Irvine, and then I had the opportunity, like I said, to move to UCR and start my new lab. These are just some old pictures with my first set of students who were helping me put together the lab and start somewhat different projects, because obviously that was a good run, but I wanted to have my own independent research program. But I had learned a lot about spray and droplet characterization. So one of the things that I also learned about was droplet surface impact and impact dynamics, and also cavitation, because we were inducing some of the cavitation bubbles in regions where we had thin layers of liquid when we were firing the laser. So I had the opportunity back in those days to host one of my then became a PhD student to work on thermocavitation. And in this case, we try to not only characterize thermocavitation, but also use it as a tool to induce intentional damage on the skin to create some pores for drug delivery. And in case, I mean, I'm pretty sure you, as mechanical engineers, you've heard a lot about cavitation. You know that most of the times that's an unwanted phenomenon, like when it causes a lot of destruction in propellers and blades. You don't want cavitation to happen there. In nature, some preys use that to sting to knock out their prey and you know, survive. And in engineering, we have also learned ways for using those cavitation bubbles and the shocking effect that they produce once they collapse to create intentional perforations on very hard materials like titanium plates. So with all this background, when Juan Pablo came to my lab, we decided we would probably look into something more oriented towards the biomedical applications that I was interested in at that time. We started, like I said, by characterizing how the bubbles would grow over time for a given laser power. We would also exert some control on this has some, you can hear that. So we were basically playing with the laser power, and we were controlling the frequency at which these bubbles would be generated within this very highly absorbing solution. And then through some simulations, we would also estimate what was the temperature field around the cavitation bubbles as they grew and collapsed, to have an idea for what was the potential damage that we could induce around the bubble formation. So here's another temperature map that shows some of the work we did in that sense. So we turned this around, and we started working with some agar gels to simulate tissue. We generated those bubbles within that gel, and then we looked at what kind of damage we were inducing. So we worked with pig skin, for example, in this set of experiments, and we also took some slices to stain them with H&E staining, which is a traditional staining procedure to determine the damage that you induce in the different tissue constituents. And we placed them side by side to see, on the one hand, how deep those drugs were penetrating into the tissue, and then correlate that with the type of damage we were inducing in pig skin. So we concluded that with the appropriate and optimal cavitation bubble number and power, we could induce enough damage to the epidermis and allow for the drugs to penetrate without inducing too much damage to the dermis, which was what we were trying to avoid, because that's where you get the permanent damage that leads to scarring and other problems. So this was one of the works we did back then, and I just wanted to show you as an example. The lab continued to grow beyond the capacity that that we first had, and I had to build another lab for more optical characterization studies. The group was growing, and we were getting interested in some of the topics that you see listed here. So I was fortunate enough to get the support of my dean back then, and we grew that. And one of the works that we started back then was the optical characterization of transparent zirconia. This is yttria stabilized zirconia. This is a project that was actually started by my colleague, Javier Garay, who's a professor at UC San Diego now. And he's a materials expert, and he was developing these type of materials with the purpose of creating a transparent ceramic, as you see in these pictures. So these, zirconia is a very traditional ceramic used for many applications, but I'm pretty sure you have never seen it in a transparent form. So using this kind of technique, which he coined as the current activated pressure-assisted densification technique, or CAPAD, he was able to induce these high heating rates and therefore decrease the processing time to retain the nanostructure of those materials and therefore make them transparent. And then through some annealing processes that he could perform after that, he could even tailor the color of those samples because ultimately what he was after was to develop optical filters. Now, again, the point was to develop a system that would make those defects decrease in size so that they were shorter than the wavelength, and therefore less scattering events happened, and light could penetrate deeper or be unaffected and therefore be transparent. Now, the reason we joined efforts is because, well, he needed some optical characterization for those materials, and that's the kind of thing that we were doing in my lab. But it also led me to think that we could do something biomedically oriented with this material, because I knew zirconia is a biomedically compatible material. And so we started working, after characterizing those samples for him. As you can see, they had, depending on the annealing time, they had some transmissivity and absorptivity that was convenient for the purposes that we had in mind. We also joined efforts with one of my colleagues who happened to be working in my home country, in Mexico, just across the border in Ensenada, Baja California, so that's not too far from where UCR was. So we could travel back and forth and go to his lab, and at that time, he had a Thai sapphire laser that he was using for materials processing. And what we did is to start working on those yttrium-stabilized zirconia materials with the purpose of writing waveguides. And you'll see why the waveguides became an important issue for us later on when I talk to you about the window to the brain. But the fact that we were able to write waveguides on those materials was a breakthrough, and it was already leading us toward the next big step. I'm going to take a side note here, because as a result of that work that we initiated back in 2010, 2011, later on, a few years, 2019, we were lucky enough to publish this paper on Science that we obviously feel very proud of, and that came as a result of the work we have been doing with those ceramics and the lasers that eventually I got from my lab, and we started doing it in my lab as well. And that was with the purpose of creating this welding of two ceramics with no material in between. So not only we had already made a breakthrough in terms of developing a transparent yttrium-stabilized zirconia material, but now we were welding two pieces of the same material without any material in between. And the way we achieved this was essentially by fine-tuning the optical properties of those ceramics with the laser parameters that we had full control on. So we had femtosecond lasers that were tunable in terms of frequency, so we could control the pulse duration and the frequency at which those pulses were fired. And we also had full control through the thermal processing of those materials in terms of their absorption and scattering. So these are just samples of the kind of experiments that we were doing, first to measure the transmission through those materials once they were annealed at different amount of hours. Here we were looking at the diameter of the melt diameter, right, and plotting that as a number of the laser pulses that we were delivering, for both high absorbing and low absorbing samples, and then also for pulses of 2 picoseconds and 230 femtoseconds. And we optimized, again, this system to show that we were getting a laser-affected zone with a microstructure that was resembling the type of structure you see when you weld two metals, right? But these were ceramics. And so we felt pretty good about the fact that these were true welding of these two materials. We obviously needed to satisfy certain standards, so we did both vacuum measurements as well as shear measurements to prove that those welds were at least surpassing the STM standards for welds, and we proved that they did. So I guess that's what gave us, or gave the folks at Science the proof that they needed to publish our paper. And it's gotten a lot of citations. Obviously, this has been a very important contribution. We continued working, or one of my students continued working on some work related to laser processing of materials. So now I was starting to drift a little bit towards that laser processing of materials. He worked on this nanostructured surface treatment known as LIPS, laser-induced pattern structures. And what we did with this is essentially look at the effect that had on the erosion mitigation. So for systems where there's a lot of cavitation systems, bubbles going on, it seems that when you have these type of pattern structures on the surface, then you have far less erosion due to the bubble formation and explosion. One step further that is still future work is to not only get these lips, but also get the hierarchical structures within the lips. So next fine-tuning of processing within those big lines that you see that were already manufactured with the laser, so that you get a smaller scale type of feature that further helps with mitigating erosion. Related to that, another one of my students was also looking at how was this influencing the bubble dynamics near those surfaces. And we proved that when, compared to when you have a bubble formed right next to a flat surface, when it does get formed with the exact same parameters near a surface that has those kind of structures and has some gas trapped in them, there you have a completely different dynamic, right? So it collapses in a different way, and it exerts different forces perpendicular to the surface. So once again, this in terms of mitigating the erosion in certain surfaces seems to be a good way to achieve that goal. Okay, so but going back to this issue of the yttrium-stabilized zirconia, I had an opportunity back in 2010 to go back to my home country. I had not been back there for professional reasons, and spent there a sabbatical year working at one of the premier national labs in central Mexico. And that's where, talking to some of the folks over there, plus all the conversations we had in the past, we realized that one of the challenges that some of the neurosurgeons had was that they were working with these type of materials when they required cranial implants, none of which is transparent, and therefore it does not provide the possibility of tracking any kind of regrowth of a tumor if that's what they removed, or any kind of inflammatory process, because they're opaque, right? So they had to basically go back and reopen the skull in order to have any kind of tracking. There were also some issues related to the infection that happens in more than a few cases, and then they have to redo the surgery. But again, going back to that material, which I knew was biocompatible, we devised the possibility of developing those implants with the ceramic, with the zirconia ceramic. So I had when I came back, I had the fortune to recruit one bright PhD student who was interested in a lot of biomedical applications, and she decided to take on this big challenge of proving if we could indeed use this type of ceramic to develop a cranial implant. And so that's what gave place to that concept of the window to the brain. That's in a cartoon, a schematic of how we devised this taking the place of a part of the skull. But here's also where those waveguides that I showed you before were already embedded in that concept. If we could ride waveguides in a direction perpendicular to the surface, the idea was that we could penetrate even deeper with our lasers. And perhaps in a third iteration of that, if we could somehow connect them to optical fibers, we could then shine light through the waveguide into the optical fiber and into the tissue, so to penetrate even deeper. Okay, so let me tell you really briefly about the first challenge that Yasmin was her name, that she took on, and it was just a proof of concept. We wanted to prove that if we actually imaged through this new material compared to the imaging we would obtain through the native skull, we would get a better image. So we first cut one of these pieces. We cut out half of the parietal bone of a mouse. We put that implant there. And then using a traditional OCT system, an optical coherence tomography, which is the light equivalent of ultrasound, we were able to obtain these kind of images. So this is the one we obtained through the skull surface versus the one we obtained through the implant. And we showed that the photon density that we were able to obtain through the implant had more contrast. And in fact, we were able to see that shaded region around there, which coincides with the location of the corpus callosum, which is one of the anatomical structures of the mouse brain at that location. So we obviously plotted these. The average intensity is a function of depth, and that's where we see that structure. So this was good enough proof that, yes, we were able to obtain better images, and that alone was sufficient motivation for us to continue. We published that in a short communication in this journal that, again, got a lot of citations because there's a lot of people working on these type of technologies. And even if this is ways away from becoming a reality for humans, the fact that you can have a material like that for some preclinical studies with animals and being able to image over a prolonged period of time was already a big improvement. Yeah, we got a lot of that press. It was fun to talk to a lot of those folks. So the other reason this became so important, and I'm going to close with this part, is that it also led to something that was very natural for us to do, and that is to get funding from NSF for a program, or through a program that is known as PIRE, Partnerships for International Research and Education. And the reason was obvious, that we had already been working in a binational way with my colleague in Mexico. We had already developed that kind of background, showing that we had the ceramic, we had some preliminary tests that were very promising, and NSF had partnered with its equivalent in Mexico, it's called CONACYT, to fund projects on both sides of the border. So we just put it all together. We made the case for how or why we needed to continue working, and it was approved. It was something they liked. By the way, sombrero, I hope you know what that means in Spanish. That's hat. But it also stood for something meaningful, right? So that was the synthesis of optical materials for bioapplications. And it was truly a research, education, recruitment, and outreach effort. So altogether, we put together that nice acronym, which I guess the evaluators liked it. So we got that funding. It was, like I said, meant to be for only 5 years. It got extended and is still active now. $3.6 million came to the U.S. side, and then about 2 more on the Mexican side for my colleagues. By that point in time, we included 2 other institutions besides the lead institution, and that was crucial actually, because one of the things that NSF was looking for is to get a lot of exchanges, student exchanges, and mobility. So with the 2 institutions on what we call the California region and the other ones in central Mexico, we had that kind of mobility at both the local level, but also occasionally across the country. And the other reason this was, I think, very powerful is that every institution really took lead on one of the aspects of the project. So they were not there simply because they would provide students or because they were necessary for an international partnership. They were really providing expertise to the project that one institution alone didn't have. Okay, so I showed you briefly what Yasmin did, which was essentially feasibility studies to prove that this would work. But then a couple of other students started around that time, and they took on a couple of challenges. One was to develop this LSI, laser speckle imaging. That's an optical technique which was aimed at trying to determine the blood flow within the vessels, especially vessels that had already been photocoagulated or somehow affected. And we wanted to see what the angiogenesis or the reformation of vessels in that region was looking like. But OCT imaging is an expensive way of achieving that kind of measurement. LSI is a very cheap one because it only depends on the light reflection from a simple light source. It doesn't even have to be a laser. And so, NAMI started working on developing that system and made comparisons with OCT to show that we could get equally valuable information that way. And then David took on essentially extending or prolonging the time through which we could have these type of implants on the mouse, on the mice, and then showed that the mice were still viable. There was no infection, there was no inflammation, etc. Down to, I forget if it's there. I'm sorry about small font, but I think we extended this past 6 weeks, which for the lifetime of an experimental mouse, it's a long, long time. The other thing David realized as we were getting ready to translate this to hopefully human use is that some of the YSE samples were actually cracking. They were cracking even at relatively low laser energies. And that was not good, right? Obviously, we wanted this to withstand laser light as it was used either for diagnostics as well as for therapeutics. And so that led us to start looking at a different material. And what you see there is a much thicker sample of something known as ALON, or aluminum oxynitride. It's a relatively new material, and it's proprietary. There's one company that manufactures that and doesn't want to give the secret away, and that's fine because we've been able to get some samples from them, and we're collaborating and running the same kind of laser tests that we did on the other ceramic, but now on ALON. So if that works well, we expect that to solve the thermal issues that we were having with Yttria, and then with Yttria stabilized zirconia, and then continue on with this material. Later on, Aditya, one postdoc, joined my lab, and he was adamant about not only continuing on with the LSI work, but actually developing a portable laser speckle imaging, so that not only we reduce the cost of the imaging we wanted to have in a surgical setting, like an OR for this type of surgeries, but also use it on a clinic setting even for things like just monitoring the healing of a wound or something that dermatologists would use for other applications. So he's been working through the support of an I-Corps grant to develop this handheld portable laser speckle imaging device. All right, so I think I have 15 minutes left or so. I want to wrap it up by telling you a little bit about the latest one of my projects. This is a large project that is part of an NSF ERC. ERC stands for Engineering Research Centers, and that's sort of the crown jewel of NSF support. This ERC is called ATP Bio, that stands for Advanced Technologies for Biopreservation of Biological Systems. And as you can tell, there's a whole variety of systems that would very much benefit from having successful technologies that are able to preserve tissues or cells or organs, whole organs, in order to maintain them in a frozen state for a while, but then rewarm them in order to make them functional again. This big center relies on 3 thrust areas. One that deals with the biological engineering aspect of it. The second one that deals with the multi-scale thermodynamics of water. So this is to understand exactly how the freezing process takes place and how to properly freeze those biological systems so that they don't get destroyed either during the freezing or the rewarming process. And the third one, which is the one I'm part of, that deals exclusively with the rapid and uniform warming of the tissues. This is, like most ERCs, has to have a lot of collaborations. There were four core institutions led by University of Minnesota, MGH, Harvard, and then UC Berkeley and UC Riverside, where I was before. We were the four institutions. Since I moved to A&M, now A&M has joined the ERC, so now there's a 15th institution. And these are the pictures of the PIs and associates that have been working on this. To talk a little bit about thrust area 3, I'll tell you that one of the big challenges we have has to do with the scale of the system that we're working with. If we have very small scale systems in the order of microliters, right? So these are typically droplets that contain cells or embryos, for example. Then we don't need to use what this arrow here represents, which is CPAs or cryoprotective agents, or we need very, very small concentrations of them because they can be frozen before they actually crystallize and form ice and break the cell membranes, right? But then what happens is we need very large heating rates in order to overcome the ice formation when we rewarm them. When we work with very large systems that are, for example, full organs, they are, in terms of volume, they are on the scale of liters. There you need a tremendous amount of cryoprotective agent, which then makes it highly toxic. So you may not be constrained so much by the warming rate, but you are definitely constrained by the concentration of your CPA. So as you see, there's different set or a diverse set of challenges that we have to face and overcome. For now, in my lab and my group has been focusing on the rapid rewarming of droplets, right? So these are small systems. We don't need to worry much about the CPAs, but we need to worry about heating them up really fast in the order of millions of degrees C per minute. In order to do that, and in order to do that in a homogeneous way, we also require the presence of nanoparticles that help us distribute all those photons within the droplet in a uniform and instantaneous way. And the way to do it is to preload those droplets with those nanoparticles, and then and freeze them, of course. And then once we rewarm them, have the appropriate size of nanoparticle, shape, so that their plasmonic characteristics are such that we have a proper balance between the absorption and the scattering that allows for the distribution of photons in a homogeneous way around the droplet. And so we've been working on that. This shows some of the preliminary work. This was not ours, but this is a good schematic of what happens when you have too much of absorption and all the photons get basically absorbed very close to the surface, versus when you have a lot of scattering and then you may distribute them better, but then you don't get absorption in a homogeneous way either. So you have to strike a proper balance. Another thing we're doing is adapting simple techniques like beam splitting, right? So rather than just shining light from one end, we're splitting the beam so that we can hit the droplet from different locations, so it warms up more homogeneously. Ariana and Carla have been actually running comparisons of how, different types of nanoparticles. Gold nanoparticles are, pun intended, the gold standard, right? That's what a lot of people have been working on or with for a long time. But titanium nitride nanoparticles, which is one of the kind that one of my colleagues developed at UCR, happens to show better properties for us, at least for this application. It has a stronger plasmonic effect, is more nonlinear. It is also, in terms of availability and cost, is cheaper than gold. And also, in terms of stability, right? When you hit a gold nanoparticle, especially if they're nanorods with high power lasers, eventually they melt and reshape into spheres, and therefore their plasmonic peak shifts. But with titanium nitride nanoparticles, nanorods, you don't get that. Doesn't matter how many times you hit them with a laser. So that's a convenient part. Crystal and Carla have also been working on the beam splitting technique that I was telling you about, showing that, first of all, again, compared to gold, we're getting a better distribution of heat when we have titanium nitride nanoparticles, but also that with the beam splitting of the nanoparticles in isolation or with clusters, we're getting a better distribution of heat. And then they've also been working on the toxicity or assessing yes, this is toxicity of the titanium nitride nanoparticles on human dermal fibroblasts, just to show that there's really nothing at the concentrations that we're using for these nanoparticles, but it's a big deal if we ever want to do this in a large-scale commercial way. They started also looking at embedding some CPAs into the droplets, even though we need small concentrations, to test the possibility of overcoming or bypassing the formation of those crystals that lead to the rupture of a lot of cell membranes. And they show that indeed we can have, with the proper laser warming, we don't have any kind of crystallization and ice formation. And these droplets didn't have any kind of biological system in it, but they've also been using the artemia or what's called sea monkeys, and they have shown that they can freeze them and then rewarm them, and they're still happily swimming up thereafter. So we're working on this and fine-tuning it, and this is work in progress. Okay, well, so that's it. I want to leave a few minutes for questions and answers. I obviously want to acknowledge there's a lot of work involved, a lot of, these are pictures of my group over the years. I guess I'm missing some after 2016, but I've had the fortune of working with a very diverse and very bright group of students, all the way from undergrads to grad students to postdocs, etc. And again, none of these would be possible without the help of a lot of financial support from various agencies and also very key collaborators. Particularly, these are the ones that were my co-PIs for the Window to the Brain project that I showed you about. Thank you very much for your attention.
Thank you for very lot of interesting topics here. My question here is about the thermal cavitation. Like, do you observe that it's the stable cavitation or inertial cavitation? How much temperature difference is needed to cause those thermal cavitation?
Temperature difference. So what we do is essentially we shine a continuous wave laser onto a system that has a highly absorbing solution. In this case, it is copper nitride, and we need on the order of 100 to 200 milliwatts in order to produce sufficient heating that induces the formation of the droplet.
This is probably more, you don't see the bubble, vibration, right? Oscillation. It's more kind of collapse after the.
Correct. Yes. So it forms, it grows, then it collapses, and then if you maintain the laser on, another one gets formed, and that's why you hear the sound, the clicking sound, because that's the formation of multiple bubbles in sequence.
I have a question. Thank you for your talk. About photocoagulation, so I understand that you have problems with absorption of melanin in the dermis.
Mm-hmm. Epidermis. Well, in the junction.
Yeah. Do you also have scattering? How much coherence do you need for the photocoagulation to occur? Do you have problems with scattering?
Yeah, the deeper you you want to go, the more complicated it is because of the scattering, right? So if you want to coagulate the upper vessels near the epidermal-dermal junction, you can, you can with patients with fair skin. The problem is when you have very dark skin, a lot of it gets filtered. So you can't really go that deep. Because of absorption.
So scattering is not so much your problem.
No. Now, didn't have a chance to elaborate more on that, but because of it, there's been techniques of optical clearing agents that have been developed over time that temporarily help with the clearing of skin. It helps with the scattering portion of it because obviously it's not zero, it is still there. So if you reduce the scattering, you get better penetration. But you still have the absorption of the melanin. There's no way to get rid of it. So.
I kind of had a follow-up. I want to say, first of all, thank you. It's always nice to hear from, Latinos and, mechanical engineering. So along with that, so I know, a lot of research being done, just, period overall. They know that there's a diverse group. Is there any initiative that solving that problem, that absorption, to be able to help people with melanated skin?
I'm not sure I got your question. So.
Do you know if there's just any initiative that the lab that you're working with is doing to be able to help different people instead of just fair-skinned people?
Yeah, I used to say, but I stopped saying it, that this is a relatively racist therapy because, yeah, it's readily accessible for fair-skinned patients, but not so much for colored patients. So one of the things that has been done is the development of optical clearing agents that help a little bit with the scattering to allow for deeper penetration. The other thing that's been done is developing, in fact, I had something to do with it, was the development of some suction cups with the idea of literally sucking the skin into that suction cup that has a laser on top and the cryogenic spray nozzle as well. And what that cup does when it sucks the skin is essentially dilate the blood vessels, right? So you bring more blood to the laser rather than trying to bring more laser light to the blood temporarily, right? You have to be careful because if you apply a very large suction, what happens is all those erythrocytes leak out of the vessels, right? So you get the equivalent of a hickey, right? Because now they're all outside the vessel, and you don't want them outside the vessel in order to induce the coagulation. You want them inside. So we were experimenting to find out what was the happy medium of the vacuum pressure and the time that you had to have the skin under that pressure to induce that vessel dilation and then hit it with the laser. So all of that helps for everybody, including patients with darker skin colors.
I was curious about the titanium nitride. I was wondering what the max temperature it was exposed to. You say it was still retaining its shape, and I was wondering what the name of the company was that was producing it.
No, the titanium nitride is something we've developed in-house. Yes, that's been synthesized at UC Riverside. My colleague Lorenzo Mangolini, he's the one who's been developing these nanoparticles for a while. As far as the temperature, I don't know if any of these plots shows you the maximum temperature. I can't see from here. 60, 65 degrees. Since we're looking at potential biomed applications, we're not going very high. But if you were interested in other types of applications where higher temperatures are needed, I don't think that's a problem. Yeah, exactly.
That's fantastic. I apologize before. I thought you said that it was proprietary.
No, the proprietary material that I was referring to is for that ALON, the aluminum oxide nitride. This one. This one. You see it? It's very thick. In fact, this is probably, I don't know, a 1/4 inch thick or so, and extremely transparent. You can see the paper underneath. It looks like plexiglass.
Awesome. Thank you.
deflection for the nanoparticle? Like the impedances of the two materials, or were you varying like the ratio of how much nanoparticle you used on each drop?
Yeah, we were actually playing with the shape, so the aspect ratio. Yeah, but there's other ways you can do it. I'm not involved with that part. I just know that they've been experimenting with different aspect ratios to try to fine tune the plasmonic response of those nanoparticles.
Yeah, go ahead.
Imaging, since you use the laser speckle to refract the plasma.
Well, it's just a matter of miniaturizing all the components, you know, the light source, the camera, all the electronics.
You still need to fix on the ground, actually, because you're looking at the correlation of the laser speckle.
Oh, well, yeah, when you take the image, it has to be fixed to something. But the way the LSI systems we know of now are pretty big and bulky, and they need to be at least in a clinical setting. What we want is to have something portable that any doctor can have in their office, right? And as long as they can fix it to some stable system, they can get the other thing is, we're not really after absolute blood flow, velocity. We just want a relative comparison with, say, healthy tissue versus healing tissue, for example. Yeah. Thank you. Thank you, Ricardo. I hope it was